linear motion, but that they mean forwards and backwards, up and down. All right, then we have the semicircular canals for rotational, for circular, or for spinning, any way you want to communicate. How they work is that they're all based upon a tube within a tube idea, meaning the inner tube contains a fluid called endolymph, which is the same composition as the intracellular fluid, the fluid found within a cell. And that should speak volumes to you, I'm hoping. That means high potassium, low sodium, low calcium, low chloride, because that's what inside of a cell is like. Now I have to have structures in cells to understand the sensation I'm experiencing. So if we look at the vestibule, there are two structures inside of it. In one region, it's known as the utricle, which is this part really right over here. And it has a structure called the macula, and that's the structure that will detect linear motion. And the cells are called the hair cells. But keep in mind, the hair cells are not neurons. They're just a cell that can stimulate a neuron. And how can they stimulate a neuron? Because they have these things called stereocilia. They're like cilia at the apical part of the cell of varying lengths. And they're stuck, like inside of a gelatinous mass. So when the gelatinous mass moves, it ends up moving the top part of the hair cell called the stereocilia. And if I move the stereocilia in a very specific direction, right, towards the tallest ones, right, that will be stimulating the hair cell. All these guys bend this way, it stimulates the hair cell. How is it possible to stimulate the hair cell? Because really, we have these mechanically gated channels in the stereocilia that will be distorted, and they're like a little jack-in-the-box. They're forced open. Potassium follows its path of diffusion into the stereocilia, down into the cell. That means I'm bringing a positive charge, making the membrane become depolarized. It's like a graded potential. Not an action potential, but it's a graded potential. And then they have to actually activate voltage-gated calcium channels. Calcium comes in, lands on a vesicle, exocytosis of glutamate, and I can activate that afferent, right, that sensory neuron going to my brain. And that's because to detect linear motion, the macula is oriented in an up and down position. If I take that same structure and now just tip it, let the stereocilia point forwards, then we're looking at what's known as the saccule region. And now I'm still bending my hair cells with the gelatinous mass going up and down. But now I have to detect linear motion. Same basic idea, potassium, voltage-gated calcium channels. And now I can detect what direction I'm actually moving in. Then, however, if we go to the semicircular canals, there are these things at the base, right, called the crista ampullarises. And they're just like a macula in one sense. They're hair cells that have their stereocilia stuck in the gelatinous mass. But now it's not like a block of gelatinous mass, it's more like a teardrop shape that spans the entire width of that inner tube, full of endolymph. And so when the endolymph pushes on the cupula, that gelatinous mass, it ends up bending the stereocilia. And if I bend toward the tallest ones, I stimulate, I detect that motion. And if I bend the other way, I detect less of that information. That's why it's for rotational motion.
So the calcium ions are still at a higher concentration
Outside. That's the only reason why they're going to be coming in. Because if they were higher inside, they'd be leaving the cell.
Right. Yeah.
Haley?
I didn't know this.
Work our way up.
Okay. So do we have these hair cells? Do we have these structures for both directions?
Yes.
So like one, if it's
So that's how you
this
tell,
way, it'll
right? Me
stimulate
bending
it.
this way versus
Yeah.
this way is the hair cells bending towards versus bending away.
Okay. But it's not. It is two different hair cells. It's like one that has the kinocilium on one side and one that has it on the other.
No. So all the same way.
Okay.
All right. The difference is that I've got three different sets. One pointed up and down, one pointed this way, and one pointed that way.
Mm-hmm.
And so even though I'm only doing this, the fluid, the other two are still moving as well.
Mm-hmm.
But they're not moving much.
Okay.
And so my brain goes, okay, I got two weak signals from these two, but one strong one from this one, and they're bending toward the kinocilium. That means John is moving in this type of direction.
Okay.
It's a summation of all three different messages Whichever one's the loudest is the primary focus. But when I bend this way, maybe it's going clockwise, this way it's going counterclockwise, all hair cells, same orientation in terms of short to tall.
He answered.
Okay,
Yes.
Haley?
So it's the endolith that makes the gelatinous material move, that makes the steering
Yep.
move. Does the
Exactly.
gelatinous material have a different name, or is it named gelatinous material?
It's called the cupula.
The cupula, for all of them?
Yeah, they're all made of the same crap,
They're
some,
all
stuff.
They're all
Right.
cupula, no matter which one. you're
Yeah,
looking at.
and all, in terms of the semicircular canals, we have cupulas. In terms of the vestibules or the utricosaccules, I have maculae. All right,
Okay, so
and
maculae,
just a normal gelatinous mass.
maculae is
It's the structure that has the hair cells and the ordinary boring gelatinous mass.
Okay, so in the maculae, it's called gelatinous mass, but in. the
Well, it's a gelatinous mass, they just give it a special name,
Canals, it's called.
the cupula.
Okay.
I mean, if you want, we can write AP books and say, please give it a special name to the vestibule, because they feel lonely. But they don't have a special name, it's just called gelatinous mass with otoliths. Yeah.
Okay. Okay.
So is everybody okay with this part? This is how hearing's gonna work, okay, so what's the yin?
I think that's just me.
That's just you.
I
You
just
say
need to
yin?
go home and study, that's all.
Well, so just think of it this way, it's the hair cell that receives all the information, and it has these things called stereocilia. And they have the ability to let potassium diffuse into the cell. More potassium means I stimulate, I detect that motion. So I've got two different structures for different kinds of motion. I have the vestibule for forwards and backwards, up and down, which is really linear. And then the semicirculars to detect circular motion. The vestibule has a macula, and the crus ampularis is found with the semicircular canals.
And you said they were graded potentials?
Yes, they are.
Okay, what exactly does that mean?
It just means that they're gonna dissipate as time passes by. They get weaker the further they travel. And really, if you don't get that, that's okay. It just automatically came out of my mouth in that sense. But graded means, remember when I did the demonstration of, I think it was actually trying Dr. Michelle, who's actually still in the same place as she was way back then, right? And I was talking and Taz could hear me, but the rest of you could not, because my voice got weaker and weaker the further I traveled. That's a graded propagation of whatever that was coming out of my mouth, which we'll get to in a moment.
But it's still enough to open the voltage-gated calcium.
Yes, exactly. So if you had very little potassium come in, you wouldn't be able to open them up. Okay, so here's the first thing about hearing, is that we're gonna create a pressure wave moving through this fluid. And the pressure wave is gonna come around the end, and it's gonna exit out through the round window. So we're gonna create the pressure in the oval window, and we're gonna relieve the pressure in the round window. We create it in the oval, and we're gonna relieve it in the round. We're looking at our three inner ear bones, right? There's the stapes, the incus, and the malleus. And then this is our tympanic membrane. And this is gonna be the external ear canal, and your auditory canal, or your eustachian tube, is found over here. So, let's look at its orientation. The cochlea is a spiral pathway, and don't really get lost trying to figure out how the purple and the orange arrows actually work, right? But it's what I just said, right? There is the oval window, right? So I'm creating the pressure wave here, and then the exiting is at the round window where the orange is. So all they're really doing is taking this and wrapping it in a spiral pattern. Don't actually get lost trying to actually figure out the path of the fluid, right? We just create the pressure in the oval, and it has to then move through my tubes, and the pressure wave will be relieved at the round window, which is right there. Well, let's look at what they did here. They opened up my spiral tubes. I've got actually three little passageways right here. So we're gonna zoom in on this, and it looks like this. It's like a Christmas tree. Again, don't get lost in it. But let's focus on this idea right here. One big collection of 3 passageways. Don't learn the names of them just yet. We're gonna take this, we're gonna zoom in, and it looks like this, right? Those are still my 3 passageways. Now, we're not going with the naming of these 2, right? But they're full of perilymph. But what I want to look at is this membrane right here, and this thing, right? This is called the spiral organ, and that is the receptive structure. It is the receptive structure for hearing. All right, so the spiral organ is the receptive structure like the maculae was and like the crista ampularis was. And of course, they're going to have hair cells. So let's zoom in on this crista, or the spiral organ here, right? And we're going to look at this bottom part here called the basilar membrane, and this other gelatinous mass called the tectoral membrane found right on top. So let's zoom in on this, and it looks like this. Right? There's your basilar membrane. Think there's a trampoline. And on the trampoline are my hair cells, and they have stereocilia stuck in another gelatinous mass, but they call it a tectoral membrane. And I can see really 2 major groups of hair cells. There's these hair cells here, and then those hair cells right there. These are called the inner, because they're more in the inner part of this structure. And there's only 1 row of them. And these are called the outer ones. And there's 3 rows of them. And to give you a better perspective of the rows, we can look at this. Looking down on top of them, right, there's my inner row, single file, but then my 3 rows together. And these are the outer hair cells, these are the inners, and the inners are what we are going to say are responsible for your, quote, hearing. The inner hair cells are responsible for your hearing. The outer ones change the sensitivity of the inner ones. So the outer ones change the sensitivity of the inner ones. Now let's go back here for a second to the previous slide. And think about, again, that gelatinous mass at the top and my hair cells with their stereocilia. What we're going to do is we're going to make this basilar membrane bounce up and down. And since the hair cells are stuck in that gelatinous mass, we're going to end up making the stereocilia move as my membrane bounces up and down. Now let's see why it's going to be bouncing up and down. This is an actual photograph. You guys can actually see what stereocilia look like in hair cells. There's your single inner row, and there are your 3 rows of outer hair cells. Each one of these collections is a single hair cell. And these things are very, very, very delicate and very sensitive. I added 2 photographs in my office hours today, so you guys don't have these, but this is what damaged ones look like. I can look at my outer row here, or my inner row, I should say. It looks a little distorted. It looks like somebody knocked some of them down. Right? There's a nice healthy set, right? And these guys look totally destroyed. And here they're completely void and missing. And they've actually missing some hair cells over here. And this can come with age, or trauma, or disease, or genetic effects. But I just want you guys to see what damaged hair cells would look like. Well, what would happen is it'll start looking a little more, sort of like, let's pick this one right here. That's stereocilia. Right? If I were to, let's say, throw a flashbang grenade in this room, right? A really loud noise, which will happen way faster than that muscle can react to keep your bones from moving, will actually cause a flattening in the stereocilia.